question too, kind of briefly, but I wasn't too clear on the correct answer, so you just get like clarify real quick.
This one here. It's, the question is, basically, I was trying to get a concept of things. The universality of what we're gonna be talking about here. Any kind of perturbation in your system, whether it's, this would be included in this, right? You wanna get away from bodily pain or something that'll cause you harm and death. So your body is meant to pick up things that disturb that, right? Whether it's a change in blood pressure, maybe a change in potassium levels in your blood, glucose levels, all that stuff, right? We're monitoring it. In this case, we have all these receptors in your body telling you things that might be sticking in your toe, maybe crawling on you, biting you and stuff like that. And we wanna get away from it. So this is the stimulus, TAC, just like blood pressure is the stimulus there. Right? That's a set point, right? Things should be, you shouldn't have tacks in your feet. You should be pain-free, right? And then you should have, your blood pressure should be at a certain set point, right? And when that's perturbed in some way, there's receptors to kind of pick that up and tell your, some kind of integrating center, right? In this case, both of these are the central nervous system, right? Most of our things are with the central nervous system. But sometimes you got a pancreatic beta cell that detects glucose levels. Simpler one. But here we're sending that afferent information into your central nervous system, right? There might be a place in your brainstem, right, that is going to get information about blood pressure. Right there, and think about, what do I gotta do with this? Oh, it's too high. I gotta go tell my parasympathetic neurons to calm down my heart to decrease your blood pressure, or relax blood vessels. And that was the response. Your effectors are the heart. In that case, the SA, over here, you stepped on a tag, afferent information goes in your spinal cord. Your spinal cord doesn't just say, it has to do something in a complicated way. You step on a tag, you lift up your foot. Your whole body's responding, right? Both of your legs are doing, your core muscles are doing something, everything is happening, right? That's why it's an integration center. It takes that one piece of information, integrates it with the whole response right here. That's the idea behind that. Ask me one thing. I could talk about it for hours. I know what your trick is.
No, I just understand.
No, good. It's good because actually probably we won't even get to the second part of the lecture today. I'll just tell you what questions I'm going to ask you. All right, so this is not on your slides, right? This is why I just want, again, I like to stress that we go over similar themes over and over again, right? Over here, in this case, this is a beta cell, one of those beta cells, okay? You don't have to write anything about it. It's not on your test, it's not on your slides, it's not on your learning objectives. But notice, and this is a resting beta cell, low blood glucose levels, right? The beta cell is meant to pick up high glucose levels. But in the absence of high blood glucose levels, basically it's got normal, your resting membrane potential is maintained by that potassium leak out of the cell. And that's not a normal leak channel. It's called a potassium ATP channel, which I'll get to in a second, but in any case, the cell is not secreting insulin because there's no signal for it to secrete insulin. When there is high glucose levels, what happens is glucose comes in, undergoes the whole ATP production, because that's what glucose is for. That ATP actually closes that potassium channel. So what happens when you close a potassium channel? What builds up inside? Besides potassium.
Say it again, please.
You stop that, you plugged up your potassium channel. What's gonna happen to the cell? Is it gonna get a change in voltage?
More positive.
It's gonna get more positive, right? That's gonna be the signal in that case, right? So that's gonna be the signal for this voltage-gated calcium channel to open, and that's what's gonna cause the exocytosis, right? So again, I'm telling you this because this is a universal kind of thing. We have a signal of some sort. In this case, the potassium channel was closed due to ATP production, but we're gonna see that a lot, right? These channels are gonna open and close, change permeability, cause an electric signal, which is gonna cause the release, it's gonna cause a physiological response, right? In that case, calcium entry, that's another common theme here. Okay, so here, we're going over communication, right? That is an electrical signal of some sort. And that's what we went over yesterday, like looking at the pathway through a neuron, like how electric signals, that's the whole point of neurons, is to have this electrical signal, right, being picked up and pass it on to something else, right? Either another neuron or a muscle of some sort, right? So we're gonna talk about this. Is there any questions about what we talked about yesterday? Like any kind of big picture, like what were we talking? Somebody said, what were you talking about yesterday? And I said, it's all about cells being excitable signals. They're able to carry electrical signals in some way, right? And pass on that information, right? And those two different types. We wanted to establish what resting membrane potential meant, right? That provided the basis for all those electric signals, right? You created this energy difference, and we're gonna use the influx of ions or outflux of ions to change the electrical signals. And here we're gonna kind of talk a little bit more, get a little more complex about how those graded signals are integrated for the most part to kind of change the signaling right here, okay? And then a little bit about, the neurotransmitters involved. So anytime you get these pathways, your nociceptor picks up something, it goes into your spinal cord, it passes it on to another neuron, passes it on to another neuron, passes it on to another neuron, right? It would be a lot faster if you picked up that signal if it was all electrical signals, right? If it was like gap junctions. But here we have all these delays. There's always a synaptic delay. And the reason is we want a chance to modify that signal as it goes up, right? We can modify the pain, so we can modify these signals through these chemical synapses, right? So it's gonna go electric, chemical, electric, chemical, electric, chemical, until whatever the end game is right here, right? And within a neuron, this is from yesterday again, right? We had the dendrites and cell bodies picking up information, right? The cell body and the axon hillock were the integrated centers, right? Where it was gonna decide whether to pass that message on. The output signal would be the axon, right? And then in this case, the target, right, was the postsynaptic neuron, right? With its postsynaptic membrane that was gonna receive the neurotransmitter. And that's kind of what we're getting into today, this postsynaptic membrane over here, right? What happens over there? Okay, so here we're going to discuss the chemical synapse, right? What happens in between, right? The electric signal here and the electric signal here. And this just goes over the steps of this, right? The action potential is coming down. It's coming down over here. That's that voltage gated channels all over here. It's moving down over here. And what it's doing is causing some kind of electric signal, right? It's not shown right here, but basically it's opening up calcium regulated, voltage gated calcium channels, right? And that's what causes these vesicles, which are filled with a particular neurotransmitter, right, to be released into the synaptic cleft right here. Right? That's that little space in between the pre-synaptic neuron and the post-synaptic. Right? Most of what we're talking about is the response that happens today in this post-synaptic neuron over here, right? Because this chemical signal, that's the neurotransmitter, is going to diffuse across that little cleft right there and then bind to its receptor. Similarly, like the hormones are flowing through the body and they found its receptor, but in this case, it's pretty localized to this one spot. The receptors to this neuron over here is going to be very particular to that neurotransmitter. So if this is acetylcholine, this will be an acetylcholine receptor. If it's glutamate, it's going to be a glutamate or AMPA receptor. If it's whatever it is, right, if it's dopamine, it's going to be like several types of dopamine receptors. And this is also the key, right, to why you might get a different response based on this might be one type of dopamine receptor or a second type of dopamine receptor or a third type of dopamine receptor. The response is going to be very different. Right? But it's going to pick up the neurotransmitter that's releasing and cause some kind of response in the postsynaptic cell. Okay? Does this all make sense so far? Is this maybe even recap from stuff you've learned before? No. So what's the postsynaptic response? Is the question. What's the cell response to that? Okay, so this is going back over here, right? The receptor, the transmitter is diffusing across the cleft right here. It binds to a receptor. But again, there's going to be some sort of response that we'll talk about in a minute, but that's the signal, right? From that the electrical signal that came down the axon, right, is changed into this temporary chemical signal. And as we'll see, it's going to relate in the electrical signal down on the postsynaptic. But the other thing, we also want to It's an important part of all this, right? If this neurotransmitter remained in there, that response would keep going, right? We don't have that refractory period, so we wanna clear those neurotransmitters out of the synaptic cleft. Okay, there's 3 ways, that's the question when you're learning objective type of things, right? What are 3 ways we get rid of neurotransmitters? I mean, number one, we could just bring it back in to the axon for recycling. And that would just be bringing it kind of going back in its own cell and recycling right here. So that'd be like a reuptake one. Reuptake, you're just bringing it back into the cell, getting it out of the synaptic cleft, putting it back in, then you can put it back in the vesicles right there. Or glial cells, we didn't talk about those, but things like astrocytes, all the glial cells that are surrounding, these are gonna be able to pick them up and then recycle them, do something else with them, right? Get rid of them. All we wanna do is clear it out of the cleft. Because we don't want that signal, normally, sometimes we do want that signal to keep going, but a lot of times we wanna clear that out of the cleft to stop the response. The other thing is there's enzymes associated with the postsynaptic membrane or the presynaptic membrane, somewhere around there. They're gonna eat that neurotransmitter up and make it inactive in some form. Alright, so in your neuromuscular junction, for instance, acetylcholine, there's acetylcholine esterase, right, that breaks down acetylcholine afterwards. And so those are 3 ways, and in fact, when we talk about this is a lot of ways. Sometimes, as I said, we do want, or some reasons, there are a lot of things that would want to keep that neurotransmitter in the synaptic cleft to kind of keep that response going, right? Some of them are bad things like poisons, and if you keep this going, you eventually like die or something for some reason. Other ones, all drugs and stuff, are kind of targeted somewhere in this area, and a lot of them are targeted at this, right? So everybody knows SSRIs. Anybody think of any SSRIs? Zoloft, all those common ones. That's a selective serotonin reuptake inhibitor, right? That is, serotonin is coming over here, it's filling in here, it's causing a response. If you don't have, for whatever reason, there's many reasons why somebody might be deficient, or maybe just more is better. This is kind of a happy pathway right here, or something like that, right? If you keep more serotonin in there, that response keeps going, right? So if you reuptake inhibitor, you take these SSRIs, block those reuptake channels, you keep more serotonin in this channel, right? And that keeps that response going at least for a while, right? So that's the idea behind those SSRIs, basically, right? You're not clearing the serotonin, which is involved in like a lot of stuff in your brain right here, from being reuptaked. Right? Does that make sense? Okay, that's another one. So a lot of drugs, in fact, right? A lot of drugs are working at any step of this pathway right here, right? And here, there's a couple ones that you see. Your effect on the transmitter production, right? The transmitter release. Botox, for instance, prevents the release of acetylcholine over here, blocking these vesicles from forming right there. And that's why works on acetylcholine, and paralyzes the muscle right there a little bit. Right there, that's one of them. A lot of them are working, basically they could be agonists of the receptor or antagonists of these receptors over here. So all these drugs, when you talk about drugs and stuff, they're always acting on synapses in some way right there, in some form or fashion. So all the above, number 37 is the answer to that. What kind of things do drugs do? Like, what doesn't it do, basically, right? And so these neurotransmitters, we can classify them in big sort of categories. Right here, the ones that are being released into there, and we can categorize them as, which in the book is labeled fast or slow, right? The fast ones, here. These are these ionotropic receptors. That is, the ligand binds, like acetylcholine. It opens up the ion channel, ions flow in, like that, right? And then you have that change in membrane potential right there, okay? Those are the fast ones. And then metabotropic ones are all those very slow ones. They're gonna be involved in G-coupled receptors, right? G-protein coupled receptors. They're going to activate that complex G protein pathway, and that's where it gets really complicated, right? And we're not going to go into any of that stuff. So when we talk about these ionotropic, we'll mention a couple of the G protein ones, but we're not going to get into the effect of them. right? So metabotropic, slow, and the inotropic ones are fast, right? That is just because they're gonna open up ion channels, the neurotransmitters, the receptors just open up, stuff flows in, changes the membrane potential. G protein has to go onto these several pathways, and you're gonna have wide, complex effects. So, again, postsynaptic responses, that is the binding of the neurotransmitter. It's been released from the presynaptic membrane, right? We're talking about at the synapse. You got the chemicals flowing down, binding to receptors, and those fast synaptic potentials are gonna open up those channels, right? And you notice those words EPSP, IPSP, that's excitatory postsynaptic potential. That is a tendency to depolarize where those, in that area of the synapse, or inhibit the incidence, that is by hyperpolarizing it. Okay, and so these are, you know, you'll see these are some of the more common ones, right? Glutamate and GABA are the big ones in your brain. Nicotine, also the nicotinic, the acetylcholine one is the one we see in the neuromuscular junction. So if I say GABA is an inhibitory neuron, what is the choice for? If I say it opens up an ion channel, what kind of ion channel do you think it might open? If it's an inhibitory one, that would call it hyperpolarization. There's two big guesses here for all your common ions.
So another potassium.
Could be a potassium one. I think that's a good guess. It theoretically could be, happens to be
It could be also CN, chlorine.
chlorine. Chlorine is the other big one that would open up. We don't really talk usually about it, but that's, GABA receptor, that's one of the most common receptors in all your brain right there. So chlorine channels, bring in more chlorine, then it's gonna slightly hyperpolarize it. It's not so different from -70, it only goes to about -63, but it is gonna hyperpolarize it. Because glutamate is opening up the sodium channel, it's going to excite it. Acetylcholine also, at least the nicotinic ones. Okay? And so whereas these ones are gonna be slow, you can see that complicated pathway we're not gonna get in. All you gotta know is it's metabotropic, causing very complex responses. And they are slow, they're slow moving, right? They're not just gonna have that immediate effect on it right there. And these are ones you'll recognize from any kind of modulation of your mood, your emotions and stuff, right? Dopamine, serotonin, all these ones are slow ones that are affect very complex ways, like other ion channels or anything like that, right? So those are the ones there. All right, so fast and slow. So if I asked you something like this, which of these might happen at a postsynaptic response? These are the possibilities. And which ones are fast, which ones are slow?
The ion channels be the fast, right?
So this would be a fast one, right? The ion channels open, is there gonna be a fast one, right? Allowing ions to enter the cell, right? The membrane permeability is altered. I didn't really get into that, but either one could be that one ultimately, but immediately the membrane permeability is gonna be altered in the fast one. Maybe it'll take a little bit while for the membrane for the other one. And then the second messenger part would be your slow metabotropic, right?
So that's what, when you say second messenger, you're talking like the G protein, right?
G protein, CAMP, all that stuff, that whole complex signal transduction thing that we talked about way early on, right? That's the pathway it's gonna have open. In a way, we're not gonna understand in this class certainly, it just gets super complicated once you start getting to those second messenger, dopamine and serotonin and stuff like that, right? So I'll give you an example. This is a brain scan over here, right? And if you put a 5-HT antagonist, a certain type of receptor, right? This is showing brain activity in a normal sort of functioning person, right? You put this 5-HT antagonist, give them that, right here. Right? All these complex pathways start opening up. There's all sorts of activity that's not normally there, right? That's the 5-HT antagonist in this case is the hallucinogenic drug LSD, right? All these pathways open in a very complex way, right? Allowing all these circuits to flow that weren't particularly flowing before, right? giving some of the weird effects. Right? So all those neuromodulators like that, right? Dopamine, serotonin, serotonin is 5-HT, right? Norepinephrine, these are all modulators, emotions and stuff like that. That's what we're not getting into. I just want to put that out there, right? But all those are going to be in this kind of complex. You could alter the state of ion channels elsewhere like that, right? But ultimately, these are going to be slow. And mostly what we're talking about is this other pathway, the fast one, and the big ones, the big neurotransmitters and receptors there are glutamate and acetylcholine, right? These are the ones that are going to have an excitatory, fast response. And then later we'll get into the inhibitory fast response. Right, acetylcholine and glutamate. Acetylcholine, notice up there it says ICR, right? An ion receptor, sodium and potassium is not specific, right? But more sodium happens to flow in. These ones are a little complex, but also they're sodium, basically, mostly sodium is gonna flow in. Those glutamate, they're gonna be excitatory to both of these, right? And you'll also notice over here you got agonists and stuff, things that act like nicotine. The reason it's called the nicotinic receptor is nicotine activated, right? So it's gonna cause that kind of response. And a lot of the other ones, agonists and antagonists, are either drugs or poisons and stuff. Okay, so here we are. And in this case, let's say this is a glutamatergic neuron sending glutamate down into this synapse right here. And this will be the postsynaptic, right, potential, right? The postsynaptic potential. And that's just a particular graded potential versus like a receptor potential, but this is all the same properties, all the same stuff. Everything we talked about yesterday with graded potentials, that is, that stimulus point of origin was the synapse. And this is where the electrical activity was generated initially, spreading out throughout the entire neuron, right? Or up to a point, right? Until it sort of diffused out of the neuron. Right? There's another thing for people, we're asking about analogies and stuff. This is one window into a big room, basically. Right? If you had a big room and you had a bunch of windows, it was very cool inside, and it was very 100 degrees outside, 70 degrees inside. You open up one window, and a lot of hot air rushes in right in that spot right there, right? And then you close the window, you can feel the temperature right around the roof, right around that window where it was the temperature change, but eventually it'll sort of mitigate around the room and it's kind of diffuse around the room, right? So it's a big cell, and this is a local change that's happening right here. This is the same conversation we had about receptor graded potentials, right? It is a graded potential. So there's a local response. In this case, we're talking about, say, glutamate or acetylcholine that is causing this depolarization, right? A wave of sodium comes in, that's causing positive charges around this area, and then it's going to kind of diffuse out. And one action potential, it'll just like go in there, cause a local response, and then kind of fade out as it moves farther away from that spot. Okay? That's all the same stuff we're talking about yesterday, except here we're just kind of focusing in on the fact that this is happening at the synapse, right? Glutamate comes down, opens up sodium channels, and then sodium flows in, for this graded response right there. Okay? So, two things can happen for that. Normally, again, like I said, most action potentials, actually any one single action potential, it will happen something like this. You should be able to understand the graph, right? If you record it, the voltage change, it goes from resting membrane potential. Sodium is flowing in, so that depolarizes, gets it closer to zero, right? And farther away, it starts to become less polarized, right? More equal to zero. But, it's going to go down over time because the channels only open up for a second. Whereas if you record it right here, right, maybe you'll see over time, you rise up a little bit, but much less than over here. And as you move away, you record it over here. And if it drops below this, it's not going to set off the action potential. That is, if the threshold, if the trigger zone was right here, it would activate it, right? So let's say, think about this, if there was an axon terminal right here causing this response very close to the trigger zone, that would have a much more powerful effect, right? The graded potential might reach that right there. But it didn't, because it's way back there. However, this one, over here, on this side did, for whatever reason, cause a strong enough stimulus to reach all the way down there. And that is, it was a very strong stimulus that was way above that, and you record it, it's a little higher. By the time it reached here, it was still above threshold, and that's where we get the action potential, right? So in other words, there's one of two things that are happening. It's gonna signal out, fade away, decay, or it's gonna be strong enough to reach the threshold region. What's that?
So no,
What? No.
not all inputs.
Right, exactly. In fact, most inputs, if that picture right there, I mean, again, like I said, only a single action potential is usually gonna result in something like this, unless it was sitting right near the trigger zone right here, right? So how do we get this strong signal right there? It's gonna be this next topic right there. Yeah. So, once that stimulus, right, gets to that point right there, right, the trigger zone, over here, it's gotta be just above the threshold. Our thresholds are above, right? 54, minus 54 won't do it. Minus 56 will do it. Minus 56, no, minus 54 will do it. Minus 56 won't. Right? So minus 50, 55, and getting closer to 0, it won't matter, right? As long as it reaches threshold, it's gonna set off an action potential. Doesn't matter how big the stimulus is. Right? It's gonna be the same exact action potential, right? Any single stimulus reaching the threshold. So how, but if it's a strong stimulus, if there's a lot of stimulus, the cell downstream, right, wants to know, why is this a strong stimulus or a weak stimulus? Because all action potential is the same. And the way we do that, the way we code for it, is to put a lot of stimuluses down there, right? Many, many stimuluses, right? So the stronger stimulus is gonna lead to more action potentials, which is gonna lead to more neurotransmitters release, which is gonna relay a stronger signal to the next cell down the line right there, right? So point is, all individual action potentials are of equal strength, so you have to increase the frequency of them, or you have to do them longer, in other words, to represent a stronger signal. And this is the coding of the strength of the stimulus right here. And then last time, in lecture 7, and we talked about that with the sensory pathway, right? You got a strong signal coming from some sensory cell in your body, right? And the way you code it is you do it much more frequently. You send a lot more action potentials down the line, and you do it longer. And that'll represent a stronger stimulus, right? That is somebody just pressing on you lightly, or somebody punching you in the face or something, right? That'll be a stronger stimulus. Okay? So stronger, more frequent, and longer will equal more neurotransmitter, right? That'll represent the signal strength right there, right? And there's two ways we can do that, right? One single neuron usually isn't gonna do it unless it was like really conveniently placed next to the trigger zone here. But if you had many neurons inputting, right, into one cell like that, then you can summate, right? You can add all those different small sub-threshold gradient potentials, and you get one big one, right? And that's how you might increase your signal strength, right? That is by kind of pooling your resources, bringing a bunch of inputs into it, and then you can get a strong enough signal to reach above threshold. Right? So 2 or more, this is called spatial summation, right? You have 2 or more neurons inputting into the same postsynaptic neuron, right? At the same time, then you could increase the strength of the signal. That gradient potential will be stronger. It won't fade away, and it'll reach the threshold, right? Or your input is going to send a lot of them. The presynaptic neuron is just going to keep on sending and never let that gradient potential die down right here. Okay. So this is the way we integrate information from these multiple neurons, right? So we're going to get into this here, right? So convergence and spatial summation. Right? This is an actual neuron. All those little blue things that you see, I'm sorry, all the little blue things you see are the axon terminals of other neurons going in, and trying to influence that neuron whether to fire. So a neuron actually could have thousands of inputs into it, either positive or negative. In this case, we're just talking about positive, right? So if one of those little ones wouldn't have any effect, right? There's constant activity there, but if all of them started firing at once, that would be a huge effect, right? And the cells, the dendrites, the cells of these neurons are all spaced up. You have these so-called spines. I'm not gonna ask about these, but the spines on a dendrite, you have the synapses and stuff, and the more little spines you have, the more synapses you have, the more connections they can have. In fact, this is the basis of memory, right? You can these are very plastic, you can grow them, you could lose them, and this is how we form stronger connections in between neurons, right? Just growing these spines. But the point is, there's a lot of ways, there's a lot of input into any one particular neuron, right? One neuron can have like thousands of input. We talked about this a little bit in those receptor fields, right? We talked about large receptor fields due to the fact that many sensory neurons were picking up a certain space, converging on one single neuron. And over here, that's going to kind of sum those together. That would be sub-threshold in the purple one, would be sub-threshold in the orange one, or the blue one. Right? But all together, they could make that fire, right? That makes sense, right? It's conversion, and it's also spatial summation. Right? So here's a neuron over here. This is one in the cerebellum, whatever, but that's the cell body. You can see those are the dendrites, right? And so these could reserve like 100,000 synapses over here. So there's a lot of input that's got to be integrated in here, and it's all going to be summed up based on the strength by the time it reaches the axon hillock, right? So all those signals coming here is going to lead out a single axon and spread that message, right? That's why the cell body and the axon hillock is like the integrating center, right? It's getting information from all over the place, right? So this is about the integration, right, of the synaptic signal. The more one neuron go into it, right, either through conversions or the location of them, right? They're going to be sending their positive signals. They're going to sum. By the time they reach the trigger zone, that's going to be enough to send the message out like that. Okay, so this is called spatial summation. Spatial summation. In this case, there's 3 excitatory neurons, right? Below each one, a little below threshold, but all together, right? They're going to create, they're going to cross that trigger zone. They're going to cross the threshold. And then an action potential is generated. Right? Makes sense? I mean, rather than that one single action potential, and any one of them wouldn't do it, but all the, a single excitation wouldn't do it, right? But all together, make 3 weak signals will equal 1 strong signal. Spatial summation. Okay, so both these neurons are here, right? These are 2 sending the same signal. And again, that was the spatial summation part. Well, there you have 3, but also you could have 1 signal. If one neuron were to have influence and one wouldn't do it, well, you just keep on sending them and keep on sending them and keep on sending them, right? Kind of like if you're sitting there, somebody says, somebody's trying to get your attention, they're like, hey, and you don't pay attention, and they hey, keep on saying that. They're like, what? Right? Finally, right? They keep on repeating the same message, same thing. And if you were sitting there saying, hey, nobody listened to you, and then 3 people started yelling at you, right? You might pay attention to it. That's the kind of idea. Like everybody's trying to influence this one, and this one I'm going to just keep on sending that message over and over again, right? This is called temporal summation because it's about a time thing. You're sending more and more signals. And the key behind this is a threshold. You have 1 over here. This is the 1 signal right over here that causes a little threshold 1. And then you wait a little bit, time 2. You send another one, there's an action potential. These have nothing to do with each other, right? Whereas you send a signal here and another one here, right? You can sum those. And that's what that means, that the trigger zone area is going to have this bunching up right here because the signals are close enough in time to sum these together. And then that crosses the threshold that leads to the action potential. Right? So the first one, right? The second one is right on top of the other one, and that causes a summation. This is temporal summation. Whereas, so these are the difference of this timing of X1 and X2 is much closer in this one. and they're right on top of each other, and that crosses the threshold. All right? And so this is just the slide on there, right? Temporal summation, they're farther spaced apart, they kind of both dissipate. They're close enough in time together, they are high enough to reach that threshold, open up the voltage gated channels, send that action potentials down, right? So we got the difference between temporal summation, spatial summation, the difference between them. Just think of that guy saying, hey. Right, so all that is summation, right? You're integrating information that is based on the frequency or based on the multiple inputs, right? And you can see that any of those are going to cause these different sort of these depolarizing events that may or may not reach threshold, right? The second thing is about integration is that some of those signals in the dendrites, in the neurons, are going to be hyperpolarizing. They're going to be hyperpolarizing, which means we're going to have an opposite effect, right? Rather than summating it, right, we're going to have to subtract potentials right here. So for these receptors, neurotransmitters, acetylcholine, right? It goes around. Acetylcholine is just one neurotransmitter, but it can bind to a nicotinic receptor, which is excitatory, or a muscarinic receptor, which is inhibitory. And the muscarinic one is more complicated. You notice over here it says G protein coupled receptor, GPCR is G protein. So it's a little more complicated, but it does cause an inhibitory response. And the other main one in your brain is that GABA right here, right? This is a chlorine channel that opens up chlorine, brings the cell more negative, right? And GABA is these GABA synapses right here. These inhibitory synapses are actually most of what's going on in your brain for some reason, right? There's much more inhibitory synapses than there's pository. And this is just a biological sort of method that is used. You could also see this in gene transcription, right? There's all this negative control over gene expression. And then all I have to do is release the negative control and the positive flow is allowed through, right? That's basically how biology, for whatever reason, finds it convenient to work, inhibiting stuff and then releasing that inhibition. Okay, so basically what's happening at those synapses, instead of depolarizing things, that is bringing it closer to threshold, you are hyperpolarizing it and bringing it farther away, making it harder for the next neuron or nearby neurons to depolarize it and bring it to threshold, right? The hyperpolarization. And it's going from -70 to -80 or -90 in these inhibitory synapses, IPSP. Okay, so they're harder to excite. That makes sense? Totally. So here's an example. Here's a certain pathway, the mesolimbic pathway. There's little neurons, they're dopaminergic neurons in your brain that project and spill happy neurons all over your brain. Dopamine over there. Let's say the feel-good pathway right here, right? Mesolimbic, everything's cool. But there's these GABA receptors, these GABA interneurons, which are spreading chlorine, opening chlorine channels, inhibiting this. And what happens when you inhibit this happy neuron over here, right? You're going to cause a decrease in dopamine release, right? Sad times, right? You're inhibiting dopamine release, and whatever this pathway was doing is being decreased because of this inhibitory pathway right here. And then also, you can inhibit this pathway over here, right? And this is what alcohol does actually. It's an inhibitor, right? It's a GABA channel also, but in this case, the particular pathway happens to be inhibiting the inhibitor of those pathways, right? And this is the complex way interneurons work. This is just kind of an example, right? You're kind of causing the stopping and release of that GABA, which again is going to reactivate this whole pathway. So there's more dopamine expressed. And a lot of drugs are going to work kind of similar. So it gets complicated. You think, oh, this is an inhibitor, but it's an inhibiting inhibitor, and that's why it gets a little complicated. You don't try to understand these pathways unless you know the particular circuitry. And don't worry, I'm not going to ask it. I'm just saying, why are there inhibitory pathways right here? It gets super complex. Right? But you get the idea here. Inhibitory pathways. Sometimes all you got to do is inhibit the inhibitor or release the inhibition, and then the pathway is allowed to kind of flow through. Here's another you know, here's the samples, study done on cannabinoid receptors. From here, this is an active inhibitory neuron. Information flow was being stopped, right? Chlorine was being pumped in, it was hyperpolarizing. Information flow through whatever circuitry this was working in was being kind of swelled. And all you gotta do, if you inhibit that interneuron right here, right, the information starts passing through this pathway right here. That's from an actual study, the cannabinoid thing, and they're looking at the effect. But that's what it's kind of doing. You're inhibiting these sort of pathways, and then information flowing in a way that wasn't before. Okay? Which explains all that. Right. So these are, again, these are super frequent, right? They're both there, you know, in this case, the ion channel's open. As you say, you know, I said, what possible channels, right? Chlorine, bringing it in, is gonna bring the membrane potential to about -63. It's usually not a big player, it's not that much, but it's enough to make it a little hard to excite. Same thing if you open up calcium, potassium channels in some way, and maybe through other more, you know, G protein or whatever. In any case, you're opening up more potassium, potassium leaves, making the cell more negative, right? And that'd be an inhibitory postsynaptic potential response right there. That makes sense? And then again, they're the most frequent ones right here. So again, I'm not gonna ask anything, don't worry too much about it, but a lot of those long-lasting ones, the slow ones, those G protein ones end up altering the state of leak channels and stuff, so you can increase the activity of potassium channels or sodium channels or something like that, right? So kind of long range activity, a little bit slower, but effect will be similar, right? But here, mostly what we've been talking about is this fast one, right? Just straight up chlorine channels or potassium channels are open, causing depolarization right in that local area. Okay, so now we get back to summation, right? That is the tendency for one or more neurons to influence the overall firing rate. Then we talked about two positive inputs, increasing it. But if you had a negative one, right, on the inside, you can cancel the other one out, right? You get an excitatory, you get inhibitory, and then basically you get nothing, the signal is not passed right there. So there's another type of summation. It's just that we're canceling the excitatory inputs through the input of inhibitory interneurons. Right? So again, two sub-threshold ones might be able to get it off right here, but now you're canceling out maybe one of them or something like that. So basically you're just canceling the input of the excitatory interneurons, right? With that hyperpolarization. And so if I said, I'm gonna inhibit this inhibitor, right? That would be, all you have to do is stop this, and then this information would kind of flow through like that. So you can do that in a global sort of way, like in this picture, that last picture, right? That'll influence the entire cell. Over here, the entire output of the cell. You can do it also individually, that's kind of like that alcohol thing with the mesothelin pathway right at the edge right here. And that'll only affect one branch. Don't worry about that so much, but there's just different ways of integrating, right? Integrating the output of this, of the signals right there. And again, if you look at something like this, right, this is just, don't worry about it, but the input into the cell, right? It's going to an excitatory and inhibitory, right? Which allows the sort of relaxation of your extensors while it's simultaneously allowing excitation of your flexor compartment there. And the knee right here, and that's like one pathway by branching off into an inhibitory one versus an excitatory one can cause these complex effects, right? Am I gonna write a question on this? No. I want you to understand basically what the whole, effect of these like divergences and pathways, all the complexities that you can get right there, right? So you can go down here, one single neuron could go branch off, and it can go to any of those, and it goes down here. And this could be maybe an inhibitory neuron that will block these, right? Whereas these ones can excite. So any kind of thing can happen right here. Same thing over here on the convergence pathway. Maybe this one is gonna ultimately inhibit this one, these are exciting. This is just the complexity of it that I'm trying to point out there. Right? And then the other thing, all these pathways could happen something like this, right? By the time this gets around, this might be over here an inhibitory one, which is causing negative feedback, allowing negative feedback to it. Right? So it's all about just integrating the pathway, right? Either summing up excitatory ones to kind of get that effect, or canceling them out with these inhibitory neurons, right? Opening up the chlorine channels, hyperpolarizing it, right? So there's the second part. As I said, I'm gonna mention a couple of things in the lab today and tomorrow that I'll have you know, and I'll give you the actual questions. I'm not gonna give you the answers, but I'm gonna give you the questions that I'm gonna ask on the test. And that way you can focus your studying directly on those for the autonomic nervous system. The only thing I'll mention is this, right, while we're here, still here for so much time. If you have questions,
Dopamine levels are increasing.
just at the end of there, we talked about the somatic. This is going to be relative to tomorrow's. The end of the somatic pathway, innervating skeletal muscles, right? This is just, since we're right here, we already talked about synapses, same kind of thing right here. Motor neuron from the spinal cord comes out, axon comes out, an action potential has been generated down the axon. It goes to a neuromuscular junction, right? This is the bottom line. This is why we have all these signaling ultimately to move muscles, right? Or glands or something like that. But targets, this is the ultimate target right here. So there's going to be a synapse, just like there was in the neurons, right? The same things are happening, basically, right? Signal comes down, right? The axon, that signal opens up voltage-gated calcium channels, right? That voltage-gated calcium channels cause the release of, in this case, acetylcholine into the synaptic cleft, into the neuromuscular junction, right? Which was filled with those nicotinic receptors. What happens there? Ions flow in, mostly sodium is going to flow in just based on the concentration gradient, and they're going to all flow in, and they're going to cause a positive response right there, in there. And almost everyone is going to trigger, every single action potential will typically trigger, in this case, in a muscular system, an action potential throughout the entire muscle, right? Because that muscle is also an excitable tissue with all those voltage-gated channels all along the length of it. They'll open up, and tomorrow we'll see what happens with that action potential signal. All right, so good. All right, so today we'll see Monday, Wednesday, people in lab.